So now we'll go into product requirements. What will the device need to do? Um, so this is another example. So you're saying the device should have a maximum setting of 2-liter volume controlled breaths at 20 breaths per minute. It shall run on battery power at a maximum settings for a minimum of 90 minutes. Shall be able to be mounted on a rolling support stand. And the ventilator and stand shall be able to traverse typical hospital door and elevator thresholds. So again, this is your product. This is what it needs to be doing. You need to validate each one of these bullets to ensure that it can work as it is intended. So now we'll go into example of maybe a verification step, a test instead of a validation test. So your product requirement is shall run on a battery power at maximum settings for a minimum of 90 minutes. So your verification test would be verify runtime on maximum settings with a fully charged new battery, and verify runtime on maximum settings with a battery that has been through 50 charge cycles. So you're testing it on a fully charged versus a half charged battery. Does it still work as it's intended? Another design specific. Then you go into your design specification. A lithium-ion battery pack rated for 100 amp hours. So verification test would be verifying battery purchase spec shows type is lithium iron, and then you want to collect and analyze test data to demonstrate battery performance over the life of the battery will meet or exceed the 100 amp hours. And let's look at another example. So you have a scenario where a gear cog that has been manufactured as per the requirements stated by the client. So if you don't know what a gear cog is, it is a rotating machine part having cut teeth which mesh with another to the part to transmit torque. So for this example, we'll see what the verification is versus the validation. So if you want to go through verification steps, you will do a comparison to design requirements of the material, dimensions, tolerance, etc., to determine if the manufactured part is within the parameters indicated by the client. To validate that, you'll assemble it in the machine is designed for and test it through the various operations to make sure it works as expected. Second line item, the act of testing and confirming that the product meets the specific requirements, not necessarily guarantee that it'll work. Then you go into your validation is the act of testing and confirming that the product can perform its intended purpose and consistently produce acceptable results. So again, this is an example, and then what is a verification? What is a validation? Another example I'm going to is blood collection tube. So laboratory testing plays a significant role in patient care. More than 7 billion tests performed each year. Test results impact over 70% of clinical decisions. Blood collection tubes are often an ignored variable in clinical laboratory testing. Components of BCTs, your tube balls, rubber stoppers, lubricants can affect the accuracy of laboratory tests. So looking at this example, you want to make sure that the issues that you're seeing, such as the components, you want to make sure that the components are validated, verified, in order for it to bring accurate test results. Again, from this example, it seems like these are often ignored. So you want to validate and verify this so that the accuracy of the test is as accurate as possible and as consistent as possible. So these are examples of potential interference from blood collection tube components in clinical testing. So going back to the example, we have these different components, and then we're going to describe how you want to validate it based off the different components. So first is a separator gel. How would you validate this? You will test to see can absorb certain drugs or analates leading to falsely reduced measured concentrations. So your rubber stoppers and lubricants, how do you test this? May leach magnesium into plasma samples, falsely shortening clotting times. Your heparin anticoagulant, variations in glucose, creatinine, calcium, magnesium, etc. Clot activators added to produce serum samples may falsely elevate testosterone levels. So you want to conduct your test to prevent the potential errors that is highlighted in order to make sure that you have a high quality product. So here is a Clinical Laboratory Improvement Act. So verification, one-time process in which the customer clinical laboratory conducts less rigorous testing to show that a manufacturer's performance claims for BCT are reproducible in a customer's laboratory environment. Validation, the process of conducting tests to prove that a device or system meets the requirements of its intended use. Laboratory personnel must validate the performance characteristics of a BCT to ensure that the BCT meets requirements for its intended use as stated in the manufacturer's instructions. Multiple analysis range of analyte concentrates over a number of days. So again, this gives you an overview of verification versus validation. So looking at a house, if you're verifying, you can verify that this is a house. But then you can validate that this house is properly installed. So you might want to validate can the windows handle certain noise levels? Can the roof handle a certain amount of water before leakage happens? So those are things you might want to validate to ensure the house was properly installed. So the two most important questions to ask: verification, am I building the right thing? And then validation, did I build the right thing? So this gives you ultimately over what we discussed, verification versus validation. So now we're going to part 6, regulatory evolution of validation concept. So in this section, we'll give historical examples of medical practices and medical strategies that give rise to the current regulatory environment concerning validation. We'll describe how the regulatory requirements evolved as a response to these strategies, and then we'll explain why guidance documents are important. So regulatory evolution of validation concept. So this kind of gives you a high-level overview of how drugs were made in old-time medical practices. There weren't a lot of warning labels. A lot of these products were just kind of just put out to the market like this, and without a lot of research, without a lot of background knowledge. And ultimately, this started to see this type of thing started to kill people because, again, there weren't any warning labels. People didn't have a lot of research. The FDA wasn't regulating these like it needed to be. So after seeing that, regulations began to evolve to protect the consumers. So because you want to ensure that patients are getting quality product and that they're not getting harmed when taking a product. This right here showing Mrs. Winslow's soothing syrup for teething and colicky babies. So again, this product was unlabeled and laced with morphine, and it killed many infants. Again, tragedies like this is kind of what helped spur the movement of having more regulations, validate, verification, because you'd hate to see somebody use a product and die, especially a young infant. So this kind of gives you a high-level overview of the regulatory evolution. So in 1906, you had the Pure Food and Drug Act that was passed. In 1936, we didn't have the Food, Drug, and Cosmetic Act. In 51 to 97, you have a bunch of different amendments that go on. And then 2000 to the present, you have a lot of continuous improvements. And even to this day, we don't have everything defined right. We have new types of medicines coming to the product, a lot of gene therapies. So the regulations are continuously evolving to adapt to the different types and new medicines that are coming to the market to help patients.